Today we're going to be hearing from Jascha. His talk will be about 25 minutes, and then there'll be time for questions and discussion afterwards. I just have to say we're recording the session, and it should be on YouTube afterwards. So yeah, you're sort of consenting to being here. And yeah, with that, let me introduce Jascha. So he did his undergrad in psychology and economics, and then moved to doing a PhD at Cambridge University. He works at the intersection between neuroscience and machine learning, which is something that a lot of our audience, or kind of what we're all interested in, I guess. And yeah, he had this preprint on spatially embedded recurrent neural networks, which we're all really excited about in the lab and are looking forward to hearing more about today. So yeah, with that, over to Jascha.
Perfect. Thanks so much. Yeah, thanks so much for the invitation. I'm very excited to share my work, or my and Dan's work. So Dan is also here with me in the meeting on spatially embedded recurrent neural networks. So this is a project we've been working on together, and it's really just to give you a bit of an overview how we got to this, is kind of a large collaboration within our department here at Cambridge, but also wider from people externally. And we really try to bring in lots of different approaches and ideas to connect structural and functional neuroscience. And I think we came to quite cool results here, which show not only how we can use modeling to kind of more formally link structural and functional neuroscience, but also just to make progress and link formal models more closely to the brain and see what kind of effects these kind of links have. So yeah, the project here, as is the title on the slides, is spatially embedded recurrent neural networks, revealing widespread links between structural and functional neuroscience findings. This is led together with Daniel Alcázar, who's here with me today, and then also DJ Strauss, John Duncan, and Duncan Astle have been involved with this project. Perfect. So let's go into this. Just to give you a bit of background where we are coming from with this project. So if we think about just normal neuroscience for now, there obviously is a lot of kind of structural neuroscience research. So we know that, the brain is a biological organ with certain constraints, and, it's hard to build and very kind of just costly to build and keep up. So there's lots of kind of structural constraints which help, which in the end of the day form the brain and, as we observe it in the real world environment. And then there's lots of functional components and functional aspects to the brain as well, right? We wouldn't have a brain if it wasn't for the function. In the end of the day, it's a super expensive organ, and it is probably strongly shaped by the point that we do have to use it to solve, problems of loads of different varieties. And this is something which in neuroscience research, kind of purely neuroscience research, but also in modeling, obviously, is one of the big components, right? Pinning down functions to different brain areas and looking at brain dynamics, these kind of things. So whilst these research communities can be seen as a bit of a, separate parts of neuroscience, where we have structural research and functional research, there are obviously these links between them, right? So we obviously assume that the structural components, in the end of the day, kind of influence the function of the brain, but also that the function then goes back and probably somehow influences the structure, right? So this is kind of an assumption which in neuroscience, I guess most people would have, and it's nearly too obvious to put on the slide, I guess. And we wanted to now go about and think about, while we all think about this obviously certain constraints, if we just do this in data, to actually find these intricate links on structure and functional, especially if they exist on multiple levels, and kind of pin them down. So we wanted to use modeling to do this. And to start off and give you an idea of what has already been done is, the modeling already has been used in order to link these kind of components. So if we look at one example where we see a link from structure to function, there's a couple of recent projects on reservoir networks where structural connectomes have been embedded in a neural network setup, and the kind of, functional effects, if you want, of this network are then observed. Two example papers here is Laura Suarez's paper and also Damicelli's paper, which we really liked and really have influenced our thinking here. Another project which then kind of does the other way around, where we look at primarily functional observations or functional optimization process and see how it influences the structure, is this now very well-known Robert Young's paper in Nature Neuro, which trained networks to solve multiple tasks and then see which kind of structure internally emerges in order to solve these multiple tasks. So this is all really cool, and we really like these kind of papers which bring together both of those aspects. But in the end of the day, we felt something really is missing here, right? So we really think, or I guess in neuroscience, we would think that the relationship between structure and function is more cyclical, if you want, right? So structure influences function, function influences structure, and it kind of goes all around. And we wanted to come to a point or arrive at a model where we can actually do this. And the reason why we think this would be really cool and get to this point is that we hope that we can use this to observe how structure and function interact with each other and shape each other. We could see which kind of complex features arise from just simple structure-function interactions. So are there brain features which you observe across brains, which kind of shape our understanding of what brains really look like, which arise from this very simple structure-function interaction? And lastly, we wanted to see whether we can then actually build a model which can show these links, but also help us understand these links in more detail. So this is what we set up to do. This is kind of the background to our project, if you want. So our approach, or the idea that we came up with, is we wanted to work on something we then ended up calling spatially embedded recurrent neural networks. The idea was the following. Let's build a recurrent neural network which is constrained biologically or structurally, if you want, while it learns to solve a task. So it's not just solving a task, but there's certain constraints, structural constraints and biological constraints built in while it's learning this task. And the idea is, if we manage to build this into the training process, that we can then observe how the brain trades off its functional goals with its structural priors and constraints, right? So the obvious first question you have to ask yourself is, what are the actual components we want to build into this model? So we wanted to start with the very first simple thing, which we've seen in lots of structural connectome research, is that the brain is heavily influenced by being in a 3D space. Generally speaking, in the 3D space, longer connections are more expensive, and hence the brain tries to avoid long connections if they aren't really necessary to solve the task. The other thing that the brain, well, while this is going on, while the brain tries to go around the very expensive spatial long connections, it is, in the end of the day, a network which wants to communicate, right? So communication has to somehow be represented in this. So while the network is trying to avoid really costly connections, it should still really care about the general communicative setup of the network, right? In the end of the day, there's lots of ways of setting up a cheap network where, in the end of the day, say, only the cells only ever talk to their direct neighbors, and no long range connections are there. So you want to have this kind of trade-off, if you want, between the 3D structural cost, but also the communicative kind of optimization here. And lastly, one thing which is very important to us is the networks that we build should still solve the task, right? So there still should be some sort of task control here, which, well, while we're doing this in a very simplified way, mirrors this idea that the brain structure is also specifically shaped by, in the end of the day, doing something. And we can't just model structural connectomes by completely ignoring the task. So the way that we then go about implement this is we start with a very basic recurrent neural network. So, the vanilla, standard, fully connected recurrent neural network, nothing LSTM or something, really just the original idea of recurrent neural networks. And we want to give them we want to implement them in a way that they represent these kind of structural and communication components. So what we do for the space is we take the recurrent neural network. So this is kind of our visualization of the standard approach here. And we assign each of those units in the recurrent neural layer location in 3D space. So we just take a box space for now. It's a very simple implementation that we did here. Assign each of those units a location in this box space, and just then take a Euclidean distance between them to tell the network how expensive are the different connections. For the communication, we rely on prior research on network communication and use something which is called weighted communicability. There's a whole kind of Loden theory behind this idea of communicability in networks. And if you come to the preprint later, we actually provide in the appendix a couple of pages on this theory view. In the interest of time for now, I'll just want to give you the intuition that we've developed is that the metric that we develop, or what we tell the units and connections in the network to do, is while you get sparse, because you try to avoid very long connections, prefer to keep around the connections which are currently very highly communicative, right? So the connections that you really rely on currently to communicate in your networks, keep those around. Everything else you can get rid of, kind of. That's we give it kind of the structural prior, if you want, towards communication. And then the task that we use here is a simple, say, one-step inference task, if you want. So basically what you have to imagine is the network sees a goal, there's a delay, then it sees two choice options, and it has to take the correct choice. So you can make an analogy here to like maze task in a way, where you have to navigate towards a goal or something along those lines. But it's also fair to just literally think about it as stimuli A and stimuli B integration, and there's a table which properly defines what the correct solution is in this problem setup. So now we have those kind of three components, and we want to bring them into networks. And it's not quite that obvious, perhaps, how one should do this. So the approach that we use is we use a regularization setup. We use regularization as a method here. So we set up our seRNNs, spatially embedded recurrent networks, as follows. So we have the kind of normal setup where we have task inputs, then we have a recurrent neural network, and we embed every unit in a box space, and there's an output. And the way that we embed this kind of box space is we set up a loss function, where the loss function is a combination of the task loss. So here is like a loss with the cross entropy, and then a regularization block where we multiply the weights by a distance matrix by a communication matrix. So these are the components that I mentioned earlier, and this basically allows us to have a network setup where the seRNNs solve a task, whilst the unit weight, while all the connection weights on the recurrent layer are constrained by the spatial distance, and also have this communicative prior in there. So this is our setup. We use the regularization setup, and I guess because the community here is a bit computational, I reckon that network regularization is quite a standard thing in the end of the day. I hope this is enough information, but obviously we can discuss more details later if that's of interest. In order to have a baseline comparison, we use normal L1 networks. So here in the L1 network, then perhaps the notation also makes a bit more sense, is the difference is we again have a loss function. So here is the loss with the cross entropy for the task. And then we just purely regularize the weight matrix, the normal standard recurrent weight matrix. So we use this as a baseline model just to, compare for the sparsity effect, right? So we need some sort of baseline model, and we just use this as a baseline model. Throughout the entire paper, by the way, we'll use the color coding used here. So we have lots of comparisons between seRNNs and L1s. seRNNs are always the red ones, and then L1 networks are always the kind of bluish turquoise ones. So the research approach, or the experimental approach we take here, is now that we train a bunch of these networks. So we start training a population of 1,000 spatially embedded recurrent neural networks, and compare them with 1,000 L1 regularized recurrent neural networks, and look for a bunch of differences which tell those two networks apart. We trained those two populations for 10 epochs. And we vary, within this population of 1,000 networks, we systematically vary the regularization strengths as so that the mean connectivity of those two populations are matched, so that the sparsity isn't just influencing the results. We try to match those populations, though the matching, just to be purely transparent here, is like one of the very complicated approaches here. We can go into more details later if people are interested in how exactly we did this. And then last but not least, we just use normal ADAM optimizations for supervised training here. Generally speaking, with all the setups and parameter choices we did in this paper and in the project that I'm going to run you through now, we always try to go with the most simple implementation that we could find. So, we try to use standard ADAM, kind of standard regularization techniques, standard initializations, and these kind of things to not influence the network setup by kind of fancy choices. And quite often we also tried different options, and it didn't generally influence the results much. So we'd always try to go with a baseline, with a simple baseline implementation. Just to highlight, or to give you a preview of where we're going with this now, is the results that we're getting out of this is that in the seRNNs compared to the L1s, we can then go and actually observe how those different implementations and priors all interact. And we show in this project that there's really cool effects which go from structure to structure function to just functional findings. And then we show that all these different findings all arise in unison, as we also kind of would hope that they're not kind of disparate findings, but they're all kind of linked together. And I'm going to now run you through the different results we have in those different sections, or at least, well, a subpart of these different results for now. So let's start with the structure here. So first of all, when we look at the structure of the networks, what the very first thing I want to point out is, now that these networks have a space, we can actually visualize those networks in a space, right? So we actually found this already quite cool, that all of a sudden we can take these networks and actually put them in a space and see how activations are flowing in this space, you know. So it's already kind of cool, and just gives us this brain-like feel to begin with. But we also just want to run a sanity check for now and see one thing we obviously expect, as it is regularization. As we regularize those networks, they should get sparse, right? And this also now gives you an idea of how those plots generally look. We have a comparison between seRNNs and L1s here, and we show that over training time, so over the training epoch, both of them kind of get sparse. So we do like kind of a total weight computation here, just to kind of see, okay, it does work, you know, they do regularize in one way or another. This obviously is totally expected, not really findings, just a sanity check. So what are the findings? If we think about brains and kind of structural components in brain, we find that obviously the brain networks have specific topologies, right? So one specific topology that we find in brain connectomes of the cerebral cortex is that they are modular. So there's kind of modular hubs, which kind of preferentially within a hub, neurons would preferentially talk to each other. So they're kind of good friends and talk to each other. And another topology, or another kind of topological characteristic, if you want, that you find is small worldness. So that means that on top of this modular structure, you also have highly communicative connections between those modular structures. So these are the first two basic structural findings we wanted to look at. And generally speaking, for all the structural and functional components, we try to go with, quote unquote, a basic finding in neuroscience, and hope that most people, well, agree it exists in the brain, but obviously none of this is without controversy, right? But as an example for today, for structure, I want to run through those two. So back to the structural view of our networks. If we now look at the modularity finding, we already see that across the training spectrum, that our seRNNs here are way more modular than the L1 regularized networks. And we also see that the modularity value that we're getting across the population on average is way closer to what we would expect for, well, then what is closer to the value we expect to see in data compared to L1. We can do the same plot for small worldness. And also here we see that the seRNNs show a higher small worldness than the L1s. Just to be explicit here, the reference value for small worldness is actually a bit kind of a difficult one to choose, because it very much depends on the size of the network and other kind of setups. So the reference value we plot in here is just the formal characteristics, if you want, for small worldness, which both of those networks, so populations achieve, so L1 and seRNNs both show small worldness. So there we already see that, okay, sparsity alone helps you to achieve those characteristics. But on top of that, the additional components we add beyond just sparsity, so in our case, the spatial embedding, which includes the normal Euclidean space and then the communication space, push the network to be even more small world. So this kind of we see is the structural findings I want to highlight. We have done a couple of more detailed analyses on generative models in these to show what wiring rules underlie these connectomes, and find that even there, the seRNNs are way closer to real brain networks. But I'll leave this for you to find out in the preprint. We want instead to move on to the structure function links. So if we think about kind of a analysis or result that we see in real brains, which are on the brink between structure and function, we often observe that the code, or that units which are close together in 3D space in brains, tend to code for the same variables. So on the left-hand side, this is a result from prefrontal cortex data recorded with MRI, where we see there's this kind of functional blips and blobs on the data here, where units which are closer together tend to code for the same thing. And then obviously across the brain, as hopefully we can all agree, there's some sort of functional modularity, at least in the kind of in the cortex of larger mammals, where there's kind of specialized areas and units which tend to be close together, tend to do the same thing. So now we want to investigate this feature which we observe in brain in our networks. And the nice thing, obviously, now is that we can actually visualize this nicely because we have those 3D visualization plots of our networks, right? So if we just take a random network from our seRNNs trained selection, which we just kind of put out and visualize, we then go and decode different variables. So as I mentioned beforehand, in our task, there's kind of well, there's a stimuli A and a stimuli B, which have to be integrated. You can call them goal and choices if you want. So this is what we do in this plot here, but also you can just think of them as stimuli A and B. And we now code here as a color, which is the kind of preferred variable a given unit has in terms of their explained variance. So this is kind of a cross-validated regularized regression that we run on this to get those values out. And just taking this random network out, if you glance over this, you already see that this is kind of hubby cluster for code in the middle, and then the choices seem to be spread out around it, basically. So there's this kind of distribution versus distributed, distributed code versus clustered code going on. Now we wanted to quantify this across the population of networks, and we did this with like a, well, permutation test if you want. So the idea is we now check, are the units which code for the same thing closer together in 3D space than you would expect by chance? And so what we will find, if you look at those results here, we find that the spatial code is really clustered in seRNNs, whilst the goal code is really clustered in seRNNs here, whilst in the L1 it's just kind of a normal even line, so it's just randomness if you want. And for the choices, it's the opposite actually. So it's really distributed, so that the kind of goal code goes around. And there's some interesting results in rodent recordings now, where we have way more data of single cells, where they also find that certain codes are distributed in clusters, and we're very excited to see, and while we're looking at more tasks and compare more tasks, to see which ones end up distributed, which ones end up clustered. Like, can we, pinpoint these features? We also then go and do a bit of visualization across the task, across a given trial. So we see that there are certain very interesting dynamics which happen between those codes. So as the trial goes on, there's really cool dynamics, and we are now working on a new approach on how can we actually quantify this information flow in the network. And one of our students is now currently working on it, and we hope to soon have really cool new quantified results on this information flow in the networks. We want to move on to functional though, right? So the last thing we want to go into here is a purely functional perspective on what those brains do, on what those networks do. So one purely functional finding that one can look at in the brain is the selectivity of cells. So if we look at the brain, what usually happens is in sensory regions, we get something like classical pure selectivity. This is kind of typical visual cortex code, which means that if a cell codes for one variable, it tends to not code for another variable. So there's an anti-correlation of the explained variances for different stimuli. If we instead look into task-solving networks, so this would be like prefrontal parietal networks, we see something called mixed selectivity, so that knowing about whether one unit codes for one variable in the task doesn't tell you anything whether it also codes for another variable in the task. Across the population of neurons, this is randomly distributed, basically. So this is kind of a specific task-solving prefrontal thing, though now researchers are looking into this phenomena in a kind of a wide variety of brain regions, and there's lots of theoretical arguments on whether or not this should be, a helpful thing, or like how this can be a helpful, very computationally effective and efficient code. So we wanted to go and study this mixed selectivity in our networks. So what we do is, first of all, we take the entire selection of networks which can solve the task, and we plot this, or we analyze this correlation between explained variances. So just to show you how this plot works is that in the middle here is the expected value. So this is pure mixed selectivity, if you want. There's a zero correlation between the two, the explained variance values across the units. And then we plot the histogram on here with the distribution, and we see that the L1s tend to be way more anti-correlated. So that means the L1s show something where if a unit codes for one variable, it tends to not code for another one. So not pure mixed selectivity, what we usually find in task-solving networks or prefrontal cortex. But if we look at seRNNs, they're way closer to this expected value of kind of mixed selectivity. On top of this, what we, actually to point this out is you obviously look at this plot now, and there's a large kind of variance across the mean here, right? So you're like, okay, yeah, cool. As a median, this might work out. By the way, the lines on here are medians. But there's so much variety left and right. Does this mean anything? In the next slide, I'm going to come back to this variance to conclude. But first, let me add one more finding. One thing which we did on the functional side as well is we looked into the unit activations. So one idea, obviously, is that these network setups should be very energy efficient. And we kind of were discussing energy efficiency in terms of resource allocation for long distance connections in the brain. So now we wanted to see whether the current setup that we have with like mixed selectivity, modularity, and all these kind of things also helps functional efficiency. So what we do is we look at mean unit activations across those networks. And basically, if you look at the plot here, you should focus on everything which is kind of 60 and below, because these are the actual regularized networks. If you want, the above here is it's a bit random. If you look at the actual regularized networks, you see that even when they're matched for the connection strength within the network, the seRNNs, quote unquote, use less energy. So they use less unit activations to implement this code. So we find this very interesting finding, and one of the reasons why we now want to move on into new modeling setups, for example, spiking networks, is that this kind of energy efficiency is one which is obviously very linked to spiking. So as a last slide here, one point that we were quite keen on is not only observing that all of these things, all of these different findings arise separately in different subpopulations of the networks that we train. Because in the brain, all of them are true at the same time, right? Modularity emerges together with small word nerve, with energy efficiency, with mixed selectivity, and all those different metrics that we study. So we were keen to see whether these also arise in unison in our seRNNs. So we did one analysis here where we looked at the entire population of networks that we trained. So these are seRNNs here, and so the population is obviously differs on terms of their regularization and in terms of their training time. And what we find that across this population, there's this one zone in the spectrum here where all of these features arrive in unison. So in this zone, we have a very critical window where networks are modular, they are small world, they're very energy efficient, they are sparse, and they show this very kind of computationally efficient mixed selectivity code. So all of this kind of arrives in unison in our model, and we see this as a very cool way of actually having a model or having a modeling setup where we can show those links between features, which otherwise, if you think about this in a purely data empirical setup, usually live in different data sets. So, structural metrics quite often live in different data sets than functional findings. And here with our models, we can actually go and provide, an experimental setup, if you want, which allows us to link all these findings together. So yeah, to conclude here, just to give you a back overview, what we think this shows, or what we think seRNNs show, is that there are lots of seemingly unrelated neuroscientific findings, which in the end of the day can be attributed to the same optimization process in those networks. So it's not just that there's, this narrative of just as one thing important and this does it all in the networks. Instead, they're all important, they're all linked to each other, and this is one kind of gradient amongst which quote unquote highly functional networks emerge. And lastly, we think that seRNNs are really cool setup to bridge between structural and functional communities. So one's obviously in modeling, but also we think this can be a really cool modeling system to bring more empirical research and link this to empirical research. And ultimately, we obviously hope that this will give us an understanding of, what's a good network setup and what allows us to train efficient neural networks. So as mentioned before, we have a preprint online. So spatially embedded recurrent neural networks reveal widespread links between structural and functional neuroscience findings is our preprint here. And we'd love for you to go read it and give us feedback. We really are keen to get a wide variety of views on this. We think this is a really cool, flexible modeling setup, which allows us to get lots of views involved. And with this, I also want to highlight Dan, who's here in the meeting with me, and we've led this project together. And this really is a collaboration of a large group of really interesting, bringing together lots of interesting ideas. If you're interested in the general, what's going on with this project, we have a website set up where the preprint is linked. We have our GitHub link there with a kind of open source implementation of this model. If you just want to play around with this, we try to put updates online there once we move into kind of neuromodeling setups. And if Marcus allows me a very last note here, if you're interested in this, obviously we love to hear from you and kind of work with you, but we are also in the process of trying to set up like an open source collaboration research community for kind of neuroscience AI interactions. So we've been talking a bit to a platform called OpenBioML, and they've been doing this for kind of biology research, biology modeling, so protein folding and these kind of things. And they've been quite interested of also moving towards neuroscience. So if you're interested in possibly collaborating, being part of like an open source collaboration on just neuroscience AI topic, we'd love for you to check this out. I linked to this on the link that's there on the slides. No promises made on whether this will ever work, but the worst case scenario, we get a cool community together and can all exchange ideas. Yeah. And with this, thanks so much for your attention. And we'd love to hear your thoughts and hear about your ideas.
Cool. Well, thanks very much for the really clear talk. I'm sure there are lots of questions, so if anyone has a question, just feel free to jump on screen and ask it, or you can put it in the chat and I can read it out. So I guess I can start. So one thing I was wondering a bit about was the way you presented it, it seemed like you have a single regularization value for maybe two or three different terms. Is that right?
Yeah, exactly.
I was wondering if you'd thought about separating that out, so you would have a different weight for each regularization term, and then you could sort of study like independently how much does each one add.
Yeah. We have thought about it. I think, well, you know how sometimes go with these projects, right? There's two alternatives and both of them are listed on a paper, and then you just go with one. Well, we thought that putting them all together is a bit easier, first of all, because it obviously removes one free parameter to control for. The other thing that we observed when we were just kind of trying different setups out, we found that actually by multiplying them all together, they kind of normalize each other a bit. So you go around a bit of normalization yourself. But I think separating them is definitely something which we want to try, especially if we add new parameters. I think that will be really important. Also, Dan has done some work beforehand on different wiring rules in generative networks. So this is purely structural networks. And we are also developing this alongside. So there he's playing around with different wiring rules, different parameter fittings, and especially ones fitting this to real data and real connectomes. I think this extra flexibility of showing, basically using this a bit as a dimensionality reduction or factor analysis, basically, where you can relate these abstract regularization features in order to show how they can arrive at different real brain connectomes is really interesting. We haven't done it here, but really keen to do it in the future.
Having said, can you hear me?
Yeah.
Yeah.
Cool.
Can you hear me, Marcus?
Oh, sorry. Yep.
Yeah. So having said that, we haven't parameterized each matrix. So when Jascha introduced it, it was one single kind of multiplication of multiple matrices. But in the last part of the preprint, while we didn't parameterize each of the models, we did train separate networks that had the different matrices involved. So we kind of removed the communication matrix, or we removed the distance matrix, for example. And you see kind of shared and specific effects, like some of the findings you can get with just one, but you can't get everything really. You need both together. So you can maybe have a little look at that, but that doesn't quite parameterize each fully, but it kind of separated it. That makes sense.
Yeah. No, okay, that was actually going to be my follow-up question, was about ablating different terms basically from regularization. Okay, cool. Now I'll have a better look at that then. Yeah, and I don't know, does anyone else have any questions? Or
yeah. Hey, so I really like the work, I guess. One thing I was thinking about is that in those two terms that Marcus was we were just talking about, it feels to me like the communication term stems a little bit less from like resource constraints or like metabolic cost kind of things. It's a little bit more regularizing towards, let's see, like a feature that we're actually aiming to see emerge in a network. Were you like, did you ever think about how this like small worldness in a sense could also emerge for something more closely related to some kind of metabolic cost as well? Something that could be a bit more like a prior, because it feels a little bit less as a prior as like the pure Euclidean based regularizer to me.
Yeah. So I'd also think about it, it's a bit of a nudge rather than a prior, maybe, if you want to change those words out. So I think this is also how we ended up thinking about it a bit. So as the network becomes really sparse, we basically nudge it a bit towards the communication. I think with the interpretation of this and whether you should see this as a prior or not, I think really depends which part of the community you come from. So I think if you're really structural, which I'm not actually, then I think you think about this more as a prior and kind of a assumed wiring rule, which underlies all of this. Whereas if you're more on the structural modeling, on the functional modeling side, then you look at this, be like, yeah, this is not really a constraint, right? So I can, and I think this really shows how having a model like this actually helps us to reason about concepts which are intrinsic to the brain and think about them. What do they mean, right? So, well, I guess this is a bit of philosophical overhead to answer your question, but I'll, more to the point. We do have a setup where we take the communication out in our supplements, and do this for the structure analysis at least, and show that they're generally speaking is the same trends, but they're just a bit weaker. With mixed selectivity specifically, I think mixed selectivity with small worldness specifically, that does depend on the communication. Again, if you're really structural, I think that we often have this feedback of, okay, that's actually a sanity check rather than a finding, which I think is true. But again, it shows also how different parts of the literature have thought about these links in a different way. Obviously, in the end of the day, they're all linked, right? And this is exactly what we show. So yeah, if with enough reasoning and thinking behind different links between different features, from communication and sparseness to modularity, all of this will be obvious, right? All of this should be logically true because, well, we think of it as true. But yeah, so I think from a structural component, it's more of a prior where it is a kind of underlying principle, if you want. From the functional component, from a functional viewpoint, I would agree. I feel about it a bit as a nudge, and my intuitive understanding is think of it as a nudge. Don't know whether Dan, you have something to add.
I think I'm probably more think of it as a little bit more of a prior, actually, more maybe it's actually on the structural side. But what becomes clear is that there's quite a few different ways that people conceptualize it. But what's most important is that the maths that basically we can really understand why it's working ultimately. So in the supplement, we've put out kind of our tentative explanation for exactly why communication, as we operationalize it as random diffusion, effectively signal propagation in the network, why, if you optimize for that in a recurrent neural network, you will likely get modular structures, and you will likely get small worldness. Effectively, you can have a look in the supplement if you're interested, but effectively what ends up happening is that if you regularize for communication, it will end up quote unquote bottlenecking weights into smaller numbers of connections. So effectively, as Jascha said in his talk, we kind of remove the redundancy, and so it kind of centers along kind of particular pathways that we pass information through. There's other ways you can think about it. If in an Adam optimizer and you wiggle around the weights to kind of go down the gradient, this is kind of doing it in a way in which we're taking the topology into account. Different people have different ways, but I think there's a lot of interesting insights into this kind of way of thinking. But yeah, you actually described it very well. And I'd expect, have a look in this up and we explain how it works. Or we can talk now if someone's interested.
Would it be right to think of it as essentially almost like a form of pruning, like activity-based pruning, or is that maybe I need to go read the supplement.
Kind of, yeah. So it's kind of, right. You can think of the weight, you can think of an L1 as pruning because it's going down in weights in one way, but it's guided pruning in some way. So it's kind of it's pruning, but taking into account the degree to which, the extent to which two regions can at all communicate their signals. So they receive signals and they propagate signals from their inputs to outputs. And so it might not always prune, it might strengthen some connections, or at least relatively prune it less, if that makes sense, depending on how you set up the architecture. In some cases, you might actually strengthen some connections and weaken your rest, or you may, exactly, just like kind of keep some along more. Because trying to make it more efficient effectively. So yeah, a guided pruning process is how we pitched it in the paper. But as Jascha said, like there's a few ways that you can consider it. Also, it changes if you, rather than starting from fully connected, you start from a growing network. All these things are interconnected and also have biological, I think, relevance as well. Really. So yeah.
Hey, I have a question.
Yeah.
Am I audible? Yeah, so you praised the constraint on topology, which was, I think, pretty interesting to me, and a lot of other things. So I think another interesting thing that we know about brain networks and in general any network that where we consider like communication is skill-freeness property. So like, did you look for it, or do you think if you somehow impose it, like you impose like how you force them to communicate with like closest neighbors, if you also force them to communicate with like nodes that are like very well connected than others, like doesn't influence the results in any way?
Yeah, I mean, it's topology, so you go ahead.
No, that's a really good question. So effectively, as you said, that in most neural networks that we observe, we see this kind of scale-free property, or at least like a log normal distribution of its connectivity effectively. You can kind of see this along the scales. It's a really good point. So in one of the analyses that we didn't cover, they actually didn't cover here, if you look in the paper, effectively we do look in the, if you mean scale-free in terms of the connectivity patterns, which is what typically people mean, we find effectively in spatially embedded RNNs that you get this nice power law distribution effect. We don't quite define, there's very specific tests on how you define these, but you find effectively like a log normal / power law, at least in these, in the distribution of the connectivity in these networks. That meaning basically you have some nodes have lots of connections and majority have very few. And they effectively distribute like that. Whereas in the L1, we don't find that to the same extent. You naturally get it a bit because of the nature of optimization in tasks. So you always get a little bit of that, but we didn't formally test for scale free, but we can see the power law distribution.
And I think one interesting thing to think about is, and we are very keen on having that discussion, is which features should actually be optimized for and which features should be arising, right? So what we are trying to do, or like I think what we are keen on ultimately, is to go down and be like, what are the most basic principles you can develop this from, and then get loads of complex stuff from it, right? So obviously you don't want to hard code everything that is true in the brain and then see, oh, it's all there, right? That's just probably not what's going on in the brain. Like not everything can be hard coded. So it's a bit of a question of what do you want to hard code. So with the scale freeness, one could definitely, put this into a network. But if it already rises, then you don't have to, right? And this is the exact idea here. Like it's a bit of a search for the most basic principles from which loads of complex stuff can arise. And I don't think we were sitting here and saying, okay, now it's solved, right? We don't say Euclidean distance communication and task is all you need. Surely not. We now set up those kind of 3 principles and are very keen to, now go ahead and, as we say in this flexible paradigm, swap in different things and, see what is needed. One very active question in this regard, for example, is that other papers you see do activation-based regularization. We do topology regularization, as you just said, basically. And those are obviously very linked, right? Minimizing activations will also start minimizing weights, because that's literally the only way of how, well, there's one of the ways how you can minimize them. So they're intrinsically related. And probably both of them are somewhat minimized in the brain. But, finding the right trade-off between what is actually optimized for is kind of the search process we are on, I guess.
Okay, thanks. And really cool paper and some really great results.
Thank you.
Thanks.
Cool. I have lots more questions, but I think maybe I should just save them. Okay, maybe I'll ask one more, and then if no one else has a question, then we can wrap up. So maybe, yeah, okay, so one very technical one which is a bit broader. I'll maybe go with my slightly broader question. So there's like more and more connectomics data around. I was just really excited to look from another preprint, also from Cambridge, which is the connectome of the fruit fly larva.
Yeah.
And I guess what I was wondering was, like, do you think there'd be any value in, like, if you, like, for example, initializing a spatial RNN with literally the positions of like a known connectome, and then working from there? I just can't really work out, like, would it tell you anything interesting, or would it tell you anything worthwhile? Or it seems like it should, but I'm not sure it does.
So I think I think we fully agree with your notion of we just can't work out whether it will tell you anything. So we've been so the space that we use here as a first space is obviously just equally spread box space. Just because we thought it's kind of the first thing that came to mind, really. Some people said we should have used a sheet, which we could have definitely done, but now we have a box. So we've played around with this idea of using, for example, locations of real human connectome, and then using that space, looking this as a basis, or that could be a fruit fly, right, to impose the distances, for example. But then the brain has some flexibility, at least, on where it places units, right? So it can move them around. So it feels like completely fixing the space is probably something that you don't want anyways. So another thing we've been toying with is whether you can somehow flexibly adjust the locations in the training process. So the very first idea, which most people suggest, is why don't you make them trainable? Which you can do, but then the brain the network just puts them all in one space, right? It all puts them like in the artificial center, and just let them all overlap. So then the next thing you do, okay, impose some sort of minimal distance between all of them, but that becomes a really cumbersome calculation all of a sudden. So now we've been thinking about maybe there's like a push-pull mechanism where you after like a strong connection kind of pulls units together, and then reconnections just can hold against it a bit, and then they get adjusted, and you start all of this up in a box and see whether they emerge something more complex, or you start with whatever we think is developmental wise the basis. Largely speaking, though, we are very interested in this whole idea of thinking about using actual brain connectomes, and I think we are very much in the thinking process of working out, is this a valuable thing to do? Well, there's definitely cool questions to answer about real connectomes, right? Like once they're mapped out in detail, to think about what is the actual information flow within them, and if you train a readout layer, which nodes does it want to grab the information from, and all these kind of things. There's obviously really cool stuff there. That's a very long way of saying we don't know, I guess. But it's a question that is also very much on the heart of the open research collaboration that I mentioned there. Lots of the ideas been pinpointed around is should we use an actual connectome, or should we use more loose priors? I don't know whether Dan has.
The thing is,
sorry, Manuel.
No, I was about to say that the thing is, it's a little bit like talking about taking an endpoint as a begin, a first point, right? I think it's actually the real goal would be closer to what you were talking about with developmental rules and writing rules, is how can we actually get there through some kind of developmental process, or algorithmic growth kind of thing, right? This is a little bit like the Holy Grail.
There's a couple of things there. So there's a really good paper from 1998 actually from Stanford that I saw actually did. They kind of did this growing pruning, and they show that they don't actually incorporate space, but they show developmental growth and proliferation. It's called computational principles of synaptic pruning or something. And they kind of show this proliferation of networks, and then pruning, actually having basically large networks that are sparse is really nice. And that time course actually gives you a lot of benefits that is afforded by that. But just to add on to what Jascha said as well, in terms of space, it's like, I thought that there was nothing in that, like there would be no, Jung in a box is the same as anyone else. But I had my Viva with Danny Bassett last week or 2 weeks ago, and actually she was suggesting that you can imagine that, if you that Euclidean matrix, the heterogeneity in edge length possibilities is quite important. So if you can imagine that, like, kind of, if you set the prior space as like a box, like a column, but with a big space on both sides, like a dumbbell, you can imagine that it would form more easily form modular network that may have evolved if the tasks were also relevant for that kind of switching or something. Like, you can imagine that basically what you get to is that none of these things are totally independent. If you get the zebrafish or fruit fly, you kind of think, what are the type of tasks, if they are specialized, and they live in ecosystems that are highly specialized to do particular repetitive tasks, what would they form their spaces to be, etc. It's kind of, it becomes a chicken and the egg thing, really, which is fascinating but also difficult. So yeah, it's one of these ones. It's like, you don't know if this is important or really profound. It's the most awkward things, but yeah.
No, that's really interesting. I mean, actually, we can talk about it in a sec, but Gabriel has this preprint with kind of like a bit like the dumbbell type network you described. So it's 2 modules and then a variable amount of connectivity between them, from fully connected, which is just one blob, to just one connection between them. Yeah, also, yeah, work in a lab with physicists, so I think they would have said you should start with a sphere.
Yeah,
Exactly.
I mean, it does. Yeah, no, I mean, to a sense, it does make sense, doesn't it? Yeah, maybe we should have done.
Or we should have done a sphere.
Yeah, probably.
Okay, cool. Well, I think with that, we'll wrap up the talk for today. So yeah, just thanks very much to Jascha and Dan as well for coming along to answer some questions. And yeah, we've got more speakers lined up in the program. It's the first Wednesday of every month. There's a link to the calendar in the chat. Otherwise, you can find them on Worldwide Neuro. And yeah, thanks very much for coming, everyone.